Hi everyone. In this first lecture, we will see what are bacteriophages. This is structure of a typical bacteriophage. They have a icosahedral head and there's a collar below the head. There's a core or a tube which is part of the tail structure. And then there is a helical sheath that surrounds the core. These T bacteriophages come under the category of complex viruses because they are neither purely icosahedral nor purely helical. The head is icosahedral, tail is helical sheath. So they are called to have a binal symmetry. And there's a hexagonal base plate, and there are tail fibers attached to this base plate, and these tail fibers are involved in the attachment of this bacteriophage to the host cell. There are two types of bacteriophages. First, virulent. A virulent bacteriophage is a bacteriophage that can infect a cell and will follow a lytic cycle, which means it will kill its host cell at the end of the cycle and will cause cell lysis. The picture that you can see representative here is the T2 bacteriophage belonging to the T bacteriophages, and it's a virulent phage which follows the lytic cycle. Left is an electron micrograph or the actual picture of this T2 bacteriophage, and this is a diagram or diagrammatic representation of this T2 bacteriophage, which is a virulent phage and follows a lytic cycle. The other type of phage or bacteriophages are temperate phages. The diagram is not shown for a temperate phage on this slide, but we are going to see the life cycle. Temperate phage is a bacteriophage that can follow lytic cycle as well as lysogenic cycle. So it is a choice between a lytic and a lysogenic cycle. Lytic cycle, as we said, is a cycle where cell lysis takes place and they kill the host cell. Lysogeny is a life cycle where the genome of the bacteriophage integrates into the host genome and replicates along with it. Let's see the life cycle of a virulent bacteriophage and a temperate bacteriophage. This is life cycle of a virulent bacteriophage such as T2, a T phage, and it's a lytic cycle. So let's see where it begins. Step number 1, the bacteriophage has attached to the E. coli. T2 phage is in fact E. coli, where the tail fiber is attached to a particular receptor site, and then the bacteriophage injects its DNA into the host cell. The entire bacteriophage do not enter the host cell, only the DNA is injected in step number 1, and this is E. coli cell and its genome. Then the enzymes coded by the phage break down the bacterial chromosome. Bacterial chromosome totally broken down. After that, the phage replicate using the bacterial materials and the phage encoded enzymes, replication of the phage genome in this step. Then the phage late genes are expressed to produce the structural components like head and tail. The heads are made, they are assembled with the DNA, tails, helical sheath, base plate, and tail fibers are made. The progeny particles are assembled, and then finally they kill the cell, cause cell lysis, and are released to infect the other cells. This is how a lytic bacteriophage or a virulent bacteriophage reproduces. Let's see the next life cycle of a temperate bacteriophage, example lambda phage. The other temperate phages as well. So temperate phages have a choice to go under lytic or lysogenic cycle. In this case, step 1, this lambda phage binds to a bacterial cell wall to the receptor sites and introduces its linear chromosome into the cell. This is a host cell and host DNA. In the next step, the lambda chromosome, which is linear, circularizes. In one of the subsequent lectures where we will see the lambda phage in detail, there we will talk about how this linear chromosome circularizes of the lambda bacteriophage. At this point, the phage has a choice to undergo lytic or the lysogenic cycle. Let's say the phage undergoes a lysogenic cycle where the lambda phage DNA integrates into the bacterial chromosome, and in the integrated state, the lambda phage is called as the prophage. Here, the lambda phage's genome is integrated into the host genome. As the bacterium grows and divides, the bacterial chromosome replicates along with the prophage. And this cycle is completed. This is the lysogenic cycle. This integration is a reversible step, and there are certain triggers that result in this prophage getting excised. Conditions, step number 6, at some point typically cause the lambda phage chromosome to become excised from the bacterial chromosome, and the lambda phage then enters the lytic cycle. During the lytic cycle, the phage encoded enzymes break down the bacterial chromosome in the previous cycle, as we have seen in case of T2, and under the direction of the phage genes, protein components of the lambda phage particle assemble, and from these circular chromosomes which have excised, linear copies of the lambda phage genomes are produced. Lambda phage particles assemble with each particle containing one copy of the linear phage chromosome, and then a phage encoded enzyme digests the bacterial cell wall, releasing the progeny lambda phages. This is the lytic cycle. In the lytic cycle, a temperate phage kills its host cell, as you have seen in this step. And in the lysogenic cycle, it doesn't kill, it just integrates into the host genome and replicate along with the host genome. So in this lecture, which was Introduction to Bacteriophages, we have seen the structure of a typical bacteriophage. We have learned there are two types of bacteriophages, virulent and temperate. And a virulent bacteriophage follows a lytic cycle. We have seen the life cycle of a virulent bacteriophage, and a temperate bacteriophage can follow either lytic or lysogenic cycle, which we have seen in this diagram. As we will see in the other lectures, more details of this virulent and temperate bacteriophages. And in this introductory lecture, we have taken example of T2 and lambda, which are both DNA phages. There are RNA phages as well, which will be one of the lectures of this course.